And if I talk for too long, you'll have to stop me.
Oh, yeah, sure. Good afternoon, everybody. I'm really sorry. My sincerest apologies because the session got delayed. There had been so many confusion about, the mixing up of the British summertime and Greenwich Mean Time. So there is apparently there is 2 times in Britain. And my apologies to Dr. Matt as well, because I haven't realized it unless, until of late. And Dr. Matt has been working in Cambridge for some time. And he has recently came in news in BBC, as well as many of the other media outlet has featured his great discovery in Antarctic algal blooms, the green snow map from space. So the Matt has done a fantastic job on mapping the green, the green algae blooms using the remote sensing at first, and then ground validation as well. So you might have read that the story has been extensively shared by several media outlets. So the story is all about this Nature Communication, this paper. The title of the paper is Remote Sensing Reveals Antarctic Green Snow Algae as an Important Terrestrial Carbon Sink. So today he's going to share the exciting story of this discovery. And along with that, this was Dr. Matthew Davey, his full name. He's working at, earlier he was with the Cambridge University. Right now also, I think he's working part-time in the Cambridge, but now he's right now he's with the SAMS, that is Scottish Association of Marine Sciences. So that is his new affiliation. And yes, the Guardian also has featured this story, so as the Scientific American as well. So yeah, it's a great privilege, Dr. Matt, and to be with you today in our blue planet. So today is World Oceans Day. So yeah. Congratulations, and yeah, I wish you the best wishes on this important day, and thanks a lot for you being in our Blue Planet series. Over to you.
Okay, thank you. Thank you for inviting me. So yes, I'm pleased I'm finally here to give the talk. Right, so I will try and attempt to share my screen, and let's see if this works. Okay, right. I think if I share this one. You have to tell me if it works or not work. Okay. What can you see? Can you see the first slide?
Perfect. Yeah. All is well. You can make it full screen. Yeah.
If I do present slideshow, how's that?
Yeah, perfect. Go ahead.
Is that better? Okay.
Yeah.
Great. So thank you again for the introduction. And yes, we're really pleased of the publication and the global audience that it reached. So we didn't expect it to be such big news. Of course, we knew it was big news, but we didn't think it'd be taken up so widely. So that's been really great. So I appreciate it is a World Oceans Day, and so a lot of this work will be on the interface between the oceans and the sea and the land. So a lot of the snow melt and the algae that we've been mapping actually end up, well, we think it ends up in the local sea. So so even though a lot of this will be talking about terrestrial stuff, it does have that link with the oceans. So yeah, so as Felix mentioned, so this I'm still part-time at the Department of Plant Sciences at Cambridge, and last month I started in Scotland at a place called SAMS, which is the Scottish Association for Marine Science, and their CCAP Algae Collection Institute as well. So I can't take all credit for this work and the paper. We're collaborating with the British Antarctic Survey with Pete Convey and Pete Fretwell and Lloyd Peck, and at Cambridge there's Alison Smith, Professor Alison Smith, who led the lab, and Monica Kralikowski and Monica Mendelova, and also Andrew Gray, who did most of the modeling and the satellite imagery work for this. So I can't take credit for any of those bits, but I'll present the overview of what we did. Okay, this is just a screenshot of SAMS, and so they're having a big World Oceans Day as well. So I think around the world, different places are celebrating World Ocean Day in different ways. Okay, so what I'm going to do over the next 30 minutes or so is talk about the background to the project, and we've been looking and trying to produce this map for about the past 6 years. So we're really pleased that it's finally out there. So I'll be talking about some of the field methodology and the data collection. I know from Felix's polar work as well, so you'll probably recognize some of these sites and so on, and the logistics involved, and then the biology, remote sensing, and what we plan to do next. Okay, so I'll just start off with what are algae. So there's two main types. There's the microalgae, and you can see some here. This is chlorella on the left and chlamydomonas on the right-hand side, and you can see that some of these actually mate. So they can divide asexually or sexual division, and they can bloom and grow in that way. And of course, the other type of algae that we've heard about earlier today is the microalgae, sorry, the macroalgae. These are the big seaweed species that we see. But most of my work is on the microalgae. And they are important. They're important ecologically because they're responsible for about 50% of the global carbon fixation. So when they bloom and so on, especially in the oceans, they do actually take up a lot of carbon dioxide from the atmosphere and so on. So they are important in terms of global carbon budgets. So most of my work, it's not looking at this huge scale in terms of what we see on the ocean. When I first went to Antarctica in 2014-15, looking at some of the vegetation, some of these snow algae blooms were, big in terms of the scale of the other vegetation in Antarctica. And you can see here at the front of the image, there's some pink on the snow, and then in the background or the midground, you can see some green. So these are algal species growing on the snow surface. And you can see a person in the middle there for scale, and then the ocean and the icebergs in the summer. So this was around about January, February in Antarctica. So this is sort of their summer season. So when you take that snow sample and you look at it under a microscope in the research lab on the base, you can see that here are the green algae swimming around. Some of them are potentially mating there as well, and there's other organisms there as well. So it's a bit of a community. If you scrape off some of the red snow and look at that under the microscope, and here I'm just going in and out of focus with the microscope, you can see the red species as well. So there's a bit of a mix. There's these small green ones and these much larger red ones. And this like little halo around the edge is what we call sort of EPS. It's like a sticky protein, sugary matrix around the cells as well, which will have some protective function. So in terms of life cycle, we're always learning, and the assumption at the time was that you'd have, in the summer, you'd have these green snow algae would be swimming around in the snow, liquid water, and they would mate. And then at some point, you would have an environmental trigger that would make them go red, and then they would overwinter in this red phase. And then in the summer, it would start again. But we now know that this very nice life cycle is probably the exception rather than the rule. So we're always learning about the life cycle of this as well. Okay, they're also very ecologically important. So here at the top, you obviously they're the bottom of the food chain as a primary producer, so they're taking up CO2. And then the picture to the left here, this is Andrew Gray. So a lot of this then melts out into the local waterways. And we know, even though we don't have the experimental evidence to show it, that we expect, this is my sort of hand wavy bit here, that a lot of krill will eat some of these species or similarly related species, and they're red krill because they take up the same, that pigment is from the algae. And of course, penguins eat krill. Penguins will then poo, and their feces can actually be seen from space. So this pigment, this red pigment, is very stable in ecology. We're also comparing it to the other small amounts of vegetation we see in Antarctica. So there's mosses and lichens and so on there as well. So we're comparing it to that. And of course, Antarctica wasn't always at the bottom of the world and a bit of a icy wilderness. Here you can see when you're in Antarctica, you quite often see these fossils of different ferns and other tree species that were there sort of millions of years ago. So it's always quite interesting when you're in this environment to come across species from the past. So we're trying to work out essentially, how much algae do we have on the terrestrial sort of marine border ecosystem in Antarctica? Can we detect these blooms from space? Who lives there in terms of a species composition, and what's the chemical composition of those species as well? And then to start off with, we're really thinking about what's the most abundant, are they the most abundant terrestrial photosynthetic organism in Antarctica? So we set off a few years ago to answer this. We've been collecting data 2015, 18, 19, and so on. And it's a mixture of satellite imagery, getting a lot of equipment from the UK via Chile down to Antarctica, doing all the measurements, and then we have field work that we do there as well, and then returning both that equipment and samples back to Cambridge for analysis. So we use remote sensing because we can't be everywhere in Antarctica. It's a big place, and if we want to map these blooms, we have to use different techniques as well as the ground validation, and then so we chose remote sensing to do this. So remote sensing, there's lots of different wavelengths in terms of nanometers, and depending on the satellite, they'll collect different parts of the spectrum in their detectors, and then we can look at that. And the satellite that we used is the European Space Agency satellite called Sentinel, and this is actually a free sort of visualization website that you can go onto. It's called Sentinel Hub Playground. If you Google that, you can then search for these different spectral bands anywhere in the world. So what happens is the satellite will fly over, go round and round the world, several times a day, and it will produce images of the ocean and in land areas as well. And you can see these like stripes of data collection on this map here as the satellite goes around. So of course, if you click on the left-hand side here, vegetation index, you'll see there's lots of vegetation in all the other continents. But if you look at Antarctica, so vegetation would be flagged up as something like yellow or blue here. It's all essentially red. So what we're trying to do is, and this was the difficult thing, is how can we actually detect these very, in relative terms, small blooms in a very white background on a large continent. So it's quite a difficult thing to do. So we set off a few years ago trying to plan this, and here's my, I always start off with a big whiteboard and trying to plan out these expeditions, and because once you're in Antarctica, you can't, if you've forgotten something, you can't go back and get it the next day delivery or something for these tubes. So you've got to plan ahead, lots of detail, and then eventually we come up with a sort of a 4-phase plan of field work, lab work, detecting these blooms from space and trying to work out how these blooms actually work and what the carbon budget is. So here are the two main sites. This is the Antarctic Peninsula. So it sort of it's like a little handle on a pan that sticks out of Antarctica, and it points towards South America. So we have Ryder Bay, which is a bit further south, way past the Antarctic Circle, and then King George Island, which is further north. So we have sort of the two extremes, or near extremes, of where we think these snow algae actually are. So when we're on the site, we actually have, we track the blooms over a season. Sometimes we go onto an island and just try to measure as many blooms as possible. And sometimes we actually have one bloom that we then, or one bloom per island that we then track over the season to see how it changes. We can then go to these blooms. Sometimes they're hidden underneath the snow. So we dig little pits. And here you can see the little green layer. It's like a cake, like a green layer, then a red layer. But quite often you'll get like a just green or just red or a mixture of the two. And I won't go all through these, but we then try to measure as many things as possible, because it's very expensive and difficult to get there. And we want to make sure that we collect as much information as we can, that we can then relate the biology to the environmental parameters that we measure. So at King George Island, this is where Andrew and Monica went. This is where they lived for a few months, doing the data analysis and collection for King George Island. It's right on the coast here, so they're quite lucky seeing whales going past on a day-to-day basis, and you can see some sort of coloration in the snow in the background. So this is Monica here doing her data collection, and you can see some of the algae already on the snow surface there. So that's King George Island. At Ryder Bay, again, here you can see different islands, very close to the sea system, and each island will have a different geology as well. And of course, Rotherham Station was on the on the right-hand side corner up there. So that's where we were based. So most of the time we were out sort of sampling. So you have to go out and do your basic ecology quadrats and taking the samples, putting them into tubes. We have data loggers around measuring temperature over the season as well. We're also trying to measure photosynthesis, and this was a bit of an experimental design. How do you actually measure photosynthesis in slushy snow? So we tried out this chamber system here that we had especially made for us by ADC, this Algae company in the UK, to try and start to measure how we actually measure the photosynthetic rate of these blooms on the snow surface. Okay, but most of it isn't actually the exciting field work. Most of the time when you're on these research bases, you are stuck in the lab doing pipetting, writing labels on tubes and looking down a microscope. So as well as the exciting images of the field work, most of the time, which people don't show you, is the lab work you have to do for hours and hours and hours after you've done your field work. So as well as the biology side for the sampling, Andrew, who's an expert on the remote sensing, he was collecting spectral images on the ground, which we can then relate to the satellites as they fly over. So these are called spectrophotometers, and we have like calibration images. We try to collect spectral images of different snow colors and snow patches and other vegetation as well. But the main problem with satellites is not a technical issue, it's actually a physics one or an atmospheric one, and that's clouds. So you can have all the expensive multi-million-pound satellite up in space, but as soon as a cloud come over, it obviously obscures your image of what you want to do. So we set out a lot of these things here. These are little calibration tarpaulins. But then, if the cloud comes over at the same time as the satellite, then you've lost your data for that day. So that can be quite frustrating. But we did get a lot of data. We were measuring green snow, red snow, lichens, dirty snow, cyanobacterial mats, all sorts of things to try to build up a database and a model for these things. So at the same time as being on the ground, so we can also do the data checks at Cambridge at the same time. So some of us who have been, well, when Andrew was in KGI, I was still at Cambridge checking the live satellite imagery to see if the data is good enough for our models. So that was quite good. So the results so far in terms of morphology and species composition and things like that. So I'll just give an overview of what we've seen. And you might have seen some of these pictures in the media over the past couple of weeks. So here you can see, try to do a panoramic shot of how some of these blooms are huge in Antarctica around these bases here. So what I want to clarify here, we're not saying that the blooms are there now because of climate change. This is the first map of, this is the baseline study, and the modeling sort of shows that maybe in the future, some of these blooms will lose some of the blooms on lower levels, but as you go higher up, some of these blooms could get bigger at higher altitudes and latitudes as well. So, so you can see some of these, these big blooms, and they are very green, they're very visible and very red. And yeah, different types. Some of them are very obvious, some of them you have to dig down, and you can see them just below the snow surface as well. So each bloom is quite different. And you can see that, you know, a huge range in morphology of species as well. And it's just gives you an overview of the size and some of the different colors that you see on these areas here. So a lot of people think Antarctica is just white and penguins, but no, it is very, very green in some parts, but that's close to the ocean. In terms of community diversity, but again, when you look down the microscope, so ignoring any sequencing of attempts at the moment, you know, you can see when you look down the microscope, you get a huge diversity in shapes and morphologies, whether it's single cell or filamentous algae as well. So that's like the green. If you look at the red, again, not all red cells are the same. They're not all this sort of nice round hematococcus type cells. You do get other species, or potentially, but probably other species as well. And this is what we're trying to identify. The first time we went there, we brought the samples back and we did some metabolic coding using the Illumina sequencing, using 16S and 18S sequencing. We can see that mainly work on algae and plants, but you know, there were a lot of fungal reads in there. There's a lot of bacteria as well. So can you hear me okay? So I had a flash up, my internet connection's unstable, but I'll keep going. Okay.
Perfect. Yeah, perfect.
Yeah. Okay. So we did a lot of metabolic coding, and we show that it's not just an algal community. There's a lot of fungi there as well. And things like chytrids, which are also important in ocean systems, as some of you might know, and a lot of bacteria. So in terms of a food web, it's something that we're trying to tease apart and try to understand a bit better. So I have a lot of interest in metabolic phenotyping as well. So we know that the environment will always influence how metabolites will change, or how the plants will respond to the environment. And so my sort of expertise and interest really is in metabolite analysis and metabolomics. And by measuring these metabolites, we can start to define the function and the purpose of some of these metabolites within the plant and how it might change under different environments. So when I bring the samples back, we're extracting the cells. We're sort of trying to break open the cells, extract all the metabolites. We can do things like gas chromatography, mass spec, HPLC, mass spec, and we do lots of multivariate statistics and trying to look at, can we see differences between red algae and green algae blooms, and so on. One of the things we did that we could actually do on base is called FTIR as well. So we're actually melting the snow melt. So these aren't concentrated samples over here. So these are just snow melt. And the snow melt, when you melt it into the liquid, actually are that red, they are that green and pink and so on. So they are very dense algal blooms as such. Then we extract them, and then we can start to measure things like the pigments, the carbohydrates, amino acids, and so on. In terms of biomass, again, at a very crude level here, you can see we're getting biomass ranging from with the red, just below 0.5 grams of biomass per liter of snow melt, sort of up to sort of over 3 grams per liter of snow melt as well, dry weight. So they are producing a lot of algae within that snow melt. And so when you extrapolate and expand this over a landscape level, you can imagine how many grams, kilograms, and tons you could have at a landscape level. And this is what we wanted to measure. So we can actually extract some of the pigments, and this is a very crude way of showing that when you extract the pigments and you do a UV vis scan, you can see chlorophyll A and chlorophyll B peaks, and the red is probably something like astaxanthin here as well. So this astaxanthin peak or compound is very important in terms of commercial interest, but also is important ecologically. With the FTIR, those of you who don't know about FTIR, so this is like a metabolite fingerprint. So it's a very quick scan of some of the major groups you might find in a cell. And you got wave number over here and absorbance on the y-axis. And so the intensity of these will change depending on the abundance of those major groups of compounds. So you can then, if you get these metabolite fingerprints, you can do things like multivariate stats, and you can see that the red blooms, for example, have high signatures for lipids and sugars and polysaccharides, and the green blooms seem to have more sort of protein, sort of amide groups as well. So we can start to see, well, actually, even if they look different, metabolically, they are different as well, and you can see that quite nicely there. In terms of flames, so fatty acids can sometimes change when things are growing in very cold environments, but generally we found that the fatty acid composition of the cells were reasonably similar. So did metabolomics called what we call MSTFA derivatization by GC mass spec. I mean, this is where we try to analyze as many metabolites as possible. And just to say that these data are in the New Phytologist paper, not in the Nature Comms paper. And again, we can do things like multivariate stats, and we can show that if you're a red bloom, you're more likely to have carbohydrates, polyols, and fatty acids being important within your cell. And if you're a green bloom, we're picking up lots of amino acids and organic acids to differentiate between those blooms. We also measured total nitrogen and carbon of the algal biomass, just to start to get an idea of what the carbon concentration is, what that carbon load is. So then we can start to work up these carbon numbers as a carbon sink and so on. Those of you interested in things like Redfield ratios in ocean systems, the carbon and nitrogen. So the published, the typical Redfield ratio for phytoplankton in the oceans and so on is 6.6 to 1 carbon to nitrogen, and we were getting 6.3 to 1. We also found that on this little figure here, as you go towards the poles, so the center of the graph where it says 0 latitude is the equator, and you go towards the poles either side of that. And we actually, so the amount of carbon isotope sort of goes more negative as you go towards the poles. And this is what we were finding there as well. So that sort of fitted the expected carbon and nitrogen signatures. So just going back to the satellite imagery now. This is, if you had a very expensive, if you had a very big budget, you could have these very expensive commercial satellites called WellView 3 imagery, and it's about $50 US per kilometer square for this imagery. So if you took a picture of one of the islands that we were working on, you can actually see quite nicely there's these green blooms and red blooms as well. Okay, but we don't have, if the government's not going to give us, millions and millions of pounds to analyze the whole of Antarctica at that level. So we have this Sentinel-2 imagery, which is free, but you can see the difference between that level of detail in a commercial satellite versus this one here. But actually, it turned out that this level of imagery was still good enough for what we wanted to do. But we also, we do lose some information, and some of that information was we can't, using the satellite or the Sentinel-2, image the red blooms as well as the green blooms. So Sentinel-2 is really, really good for the green blooms, but we couldn't differentiate or model well enough for the red blooms. So we're just concentrating on the green blooms. So we then, so now we have the ground validation work. We knew a lot about the carbon, about the photosynthesis. We had the spectral images from the ground. We had spectral images from a satellite. Andrew can then go through, he made this sort of model to then go through past images, and you could actually detect where these blooms were along the whole of the Antarctic Peninsula. So this gave us, the very first sort of model or map of where these blooms are, the intensity, because the intensity of the blooms we could relate as well, how much biomass and how much carbon are in those blooms as well. And because we have this nice latitudinal difference, Andrew then went to the sort of climate change models for Antarctica, and just to see how that might change and where these blooms might sort of change in the future in Antarctica. And that's where this sort of climate change angle came from that as well. So we actually know that, or the model was suggesting that we might lose some of these very low sort of coastal blooms. They might melt out completely, but we might start to see much larger, denser blooms further up in altitude. So the net gain, so there'd be more net gain in these blooms in the future rather than a net loss. So we know that, so at the end of the day, we managed to detect so far about 1,300 tons of green snow algae, covering nearly 2 kilometers square across that peninsula, and it's nearly about 479 tons of carbon. We also could get other information about, 6% of these blooms were within 5 kilometers of a penguin colony, and the single bloom areas and frequency decreased with lower latitude. So if there is a latitudinal difference as well. We expect this to be an underestimate because some of the algae will be under the snow that we can't detect, and we know that there's red and orange blooms as well that we can't detect as well. Okay, so what we want to do next, we want to actually do a bit more ground validation. This is what we hope to do. We hope to do it this year, but we couldn't. We probably can't do it next year, but we'll just have to wait and see. Is do a bit more of a transect across that peninsula to see, are these blooms actually there where we modeled and expected them to be. We're also doing some citizen science work as well, because this is a bit of a tourist ship route as well. So we've been asking people, tourists, and other researchers and staff in Antarctica that if they see these blooms, to take a photograph, GPS tag it, and email it to us. So we're trying to get a lot of information from other people working in the area as well. So we wanted to, so what we want to do next is really, what's the whole area of green and red blooms across the whole of Antarctica, not just that section of Antarctica. Look into a bit more about the species community composition. Be doing more sequencing and metabolite analysis to see how the algal blooms, or the algal species, are related to other algal species around the world, especially these snow algal species. Looking at things like what we call phenotypic plasticity. How are there temperature limits of where and when these algae can actually bloom? So if we know that, that will give us more information about whether they can actually survive and where they can survive in future climates. And we also wanted to do things like hyperspec drones. So rather than just relying on satellite imagery, if we have drones that obviously aren't affected by clouds, we want to sort of start looking into the hyperspec drone sample system as well. So thank you ever so much. There's lots and lots of people to thank at Cambridge and at BAS. And thank you for inviting me to talk today. And I'll finish off with a penguin. And thank you ever so much. And I'll take some questions.
Yeah, thank you so much, Matt. You may exit the window now, the presentation window. Fantastic. Okay. It is really great to have your talk in this series, and really glad to see the behind the curtain, the story of this paper. We were all excited to hear what is actually happening here. So we are actually flooding with lots of questions. So I'm really happy to see a lot of participants, despite that the talk started quite late. Apparently, my own student, Casey Saini, is asking a question. We found Cocomixa species at Larsemann Hills in eastern Antarctica. Did you find this in your data? And he's also adding, how are these algae live during the extended polar winter period?
Yeah, so in terms of Cocomixa, so the sequencing we've done so far, we've found species that are related to Chlamydomonas, Chloromonas. Interestingly, it didn't come up with Cocomixa. But when you look at the morphology, it looks like Cocomixa. It actually is calling it another Chlamydomonas species. So whether NCBI are reporting their sequencing on a different species, this is what we need to tidy up. So we have a master's student, Margot, who's been looking at this sort of tree of life with these snow algae. So so it's quite fresh data, but it's interesting that where it's putting it in the phylogenetic sort of tree. There's my gut feeling that it's going to be Cocomixa rather than this other Chlamydomonas psyllid.
Matt,
I can't even pronounce it. Yeah.
Yeah, you're still in your presentation, so you made.
Am I? Oh, okay. How do I get rid of that?
So you can say stop presenting something.
1 second. It says meeting controls. Okay, maybe it's this one. No.
Meeting control on the top, perhaps. Exit the presenter window or something.
Oh no, that will leave the meeting. So what can you see? Yeah, I can't find that. It seemed to have disappeared.
Yeah, doesn't matter. So
okay.
Anyway, let's go.
I've got it. Yep.
Perfect.
Yeah.
Yeah.
So you can just see me now. Yeah.
Yeah.
So hopefully, I think looking at the literature, it might come out as Cockamixer, but we need to do a bit of investigation work about that. But it's, so at the moment it's coming out as a species that's very similar to Cockamixer, but we need to be absolutely sure before we publish that data. Yeah.
And
what's the second question? Yeah.
Second is how do the algae live there in the extended polar winter period?
Yeah, good question, and we don't know. it's a simple answer. It's very hard to set up experiments to track these things. And well, one of the experiments I'd love to do is some sort of visual tracking of these things in the overwinter, because we know some of them will overwinter. We know that they can survive in the -20 in the dock for about a year, because we've had samples in the freezer that we can actually revive after 1, 2, or 3 years. So we know they can, even in the green phase, grow or be regenerated after that. The red cells, again, they're very good at sort of being reactivated after long periods of cold and dark. So yeah, but there's still questions on how are the blooms reseeded? Are they coming from algae that are there from previous snow that's melted out and they bloom again? We know that some of the algae are in the rocks and they could actually swim through or potentially bloom from the rocks from previous years as well. Or are they blown in from the sea, or potentially birds as well? Lots of questions.
Yeah.
Still unanswered. Yeah.
Very interesting, curiosity-driven questions. So yeah, I hope the science can. We will see an answer to it, right? In the coming days. So
yeah,
coming to.
So yeah, there are blooms in the Arctic and in alpine areas as well.
Right.
Where you go, where there's sort of snow and high altitude, you see these snow algae communities. So you can actually study them elsewhere in the world. But the Antarctic one is slightly different because you don't have the other vegetation around it that might influence that. Anyway, sorry, yeah, you're about to ask
over to
question.
Dr. Punita. Punita is here, right? Dr. Punita, over to you.
Yes, sir.
Could you
I'm
please?
here, sir.
Yeah, go ahead.
Yeah. Hello, Matt. It was a very wonderful presentation, I must say. And the exhaustive work behind this paper, I was seriously surprised. Nothing you have left: remote sensing, instrumentation, the techniques that you have used. It's simply wonderful. Because
Thank
what
you.
I believe here in India is simply like, when we are doing remote sensing studies, we corroborate it with field-based studies, but somehow that gap is there, like between pure sciences and this remote sensing. I feel that we have a huge gap here that we have to fulfill. But somehow your paper, that was very inspiring for us. Maybe we can look into this lacuna here and work out sometime. Wonderful presentation, sir.
Yeah, if we can help anyway, do contact me and send me an email.
Sure, sir.
Happy to help.
Sure.
Yeah, but definitely.
Thank you so much, Matt. Yeah, thank you.
By imagery, it looked like there's a snow algae bloom, but when you go to the field site,
yes,
it wasn't. It's actually stuff. It's actually dust, or it's
exactly,
yeah,
sir.
things that look like
you used for ground truthing and all. Awesome work, sir.
Yeah, and it's quite nice to link it to the biology as well. So yeah.
Yes, sir. Yes.
Yeah.
Thank you so much.
Okay, thank you.
Over to Felix.
So, yeah, one Mr. Anita Kirolia, she's asking a question: what types of algae exist in Antarctica during summer and winter? Well, it's kind of non-specific question, but Matt covered some of the algae that he has found, right? Yeah.
So could you repeat the question?
The question is, what types of algae exist in Antarctica during summer and winter months? Okay,
yeah. We don't know. Well, we assume it's the same species. So at the moment, it's looking like there's two main species that are there. And from the 18S, or sorry, the RBCL sequencing we've been doing, it looks like it's the same species from the 2 field seasons that we've had. So we don't. When we were there and doing the field work, sometimes we'd see a green bloom, and then at the start of the season, then later on the snow surface, you would see a red bloom suddenly appear, which is probably a different species altogether. In terms of a time sequence, yeah, I guess it seems that other different species are blooming at different times, depending on the environmental conditions and the wind direction and so on. But so it's, yeah, so I think that you can have the same species will be there during the summer and winter, but like any community and ecosystem, you'll have some species coming in at different times and blooming and so on. But this is what we're starting to learn and tease apart from the data. You know, when are we seeing consistent patterns in when these blooms are occurring or not? And how are they actually related to the other blooms we're seeing around Antarctica? So I think that there are other bases in Antarctica that are also giving us some information about their blooms and when they bloom as well. I think that this year has been lots of red blooms, apparently. So we have lots of fantastic photos of red blooms.
Great.
Yeah.
Fantastic. So I will pick one more last question because Matt is already committed only for 30 minutes. So we have a lot of questions here.
I enjoy.
Divya Bisht is asking a question. Divya's question is, as algal bloom or as a result of nutrient deposition, what is the source of these nutrients in snow which lead to the growth of these algae?
Yeah, no, good question. We did a nitrogen, a total nitrogen and carbon analysis. So we did an elemental analysis, and from that you can get an isotope signature. So the isotope signature from the nitrogen that we found in there implied that the nitrogen was coming from an animal source. And we know that near some of these blooms, there were penguins and seals and birds. So the poo, the feces from these things were probably being either wind blown or distributed somehow. So because from that isotope signature in the nitrogen analysis, it implies that nitrogen was actually coming from an animal source, which in this case would be the feces. So, but of course, you know, they also need, like any other sort of plant or photosynthesizing organism, they need phosphorus and potassium and micronutrients and so on. And we assume that they can get those from there as well, but we didn't have the analytical chemistry set up to do that on the site. So we know that chances are it will come from some animal source, but these things can actually bloom in places where there's no animals. So again, that's a good question. Where are they getting their nutrients from? Is it from the small amounts of nutrients you get from snowfall or from the liquid water? But yeah.
Matt, it's really a great pleasure to have you here today, and I hope you very best. You did a fantastic job, a first-class research. Really happy to have you here with us today. And when are you planning to go to Antarctica next?
Hopefully in January and February, but with the COVID, we don't know.
Very nice.
Yeah, so we'll plan. You know, we'll keep planning. I mean, if we can go down, it would be great. If not, we'll just have to rely on maybe more satellite imagery and keep a check on the satellite data rather than the field work. So yeah. But
you did a great, perfect. Yeah, it's a great combination to have you and Dr. Punita also. Thanks, Punita. And by the way, I didn't introduce her. She's also working on the GIS. You know, the remote sensing extensive. She's working on this field. So yes, glad to have you here, Dr. Punita. Would you like to add some points?
Yeah, sir, I spoke to him. That is what I wanted to say. Like here, when we work in remote sensing, we mostly, you know, tend to focus just on remote sensing part. But somehow when the comprehensiveness that he has, you know, explained in his studies, that is what I was very much impressed by it. So let's hope that will help us.
This is something. Yeah, something like you know, the drug paper, the combinatorial chemists. You know, they actually find something like in silico docking, but they don't actually validate with the clinical trials. So you know, you are actually doing the same thing: validation, ground level validation, plus the remote sensing. I remember, Matt. I hope you also remember when the ozone layer was first detected. You know, it was actually it was basically remote sensing said that there is an ozone layer hole. But people thought that it's an outlier, and they thought that it wasn't correct. Then it was only the British Antarctic Survey did a great job in with the remote, the ground level validation of the ozone hole. Yes, so I think the validation is extremely important, isn't it?
Yeah, definitely.
Great to have you here. Thank you.
Thank you. Thank
Have
you
a
for
nice
the
day.
invitation.
Yeah.
And
thank
you. Okay. Happy
Bye.
World Oceans Day. Okay. Thank you.
Thank you. Bye.